Assalamu alaikum warahmatullahi ta'ala wabarakatuh. I'm Salma Al-Mahdi, and today we are going to discuss metabolism of xenobiotics. Our objectives are to define the term xenobiotic, to list the classes of medically relevant xenobiotics, to discuss the significance of xenobiotic metabolism, to list the mechanisms of xenobiotic toxicity, to list factors that affect the relevant enzymes of xenobiotic metabolism, to discuss phase 1 and phase 2 reactions of xenobiotic metabolism, and to discuss cytochrome P450 in relation to xenobiotic metabolism. Xenobiotics, term-wise, in Greek, xenos means stranger. So, xenobiotic is a compound that is foreign to the body. Medically relevant classes of xenobiotics include drugs, chemical carcinogens, pollutants such as polychlorinated biphenyls, insecticides, pesticides, as well as petroleum products. The significance of biomedical significance of xenobiotics in general is relevant to so many disciplines and fields, including pharmacy, pharmacology and therapeutics, toxicology, cancer research, and drug addiction. The problem with xenobiotics is that many are highly lipophilic, so they can accumulate within the cell. The cell is a hydrophilic compartment or environment, and over time this could interfere with cellular functions. And the body figured a solution by converting those lipophilic substances to more polar forms, and this will facilitate the excretion. Xenobiotics elicit different responses in the human body. The effect could be a pharmacologic effect, a toxic effect, immunologic effect, or carcinogenic effect through three mechanisms. Xenobiotics has the ability to bind to macromolecules such as DNA, RNA, and protein and interfere with the functions of these macromolecules. Xenobiotics can alter antigenicity. For example, of a certain protein, it will act as an antigen, while the xenobiotic itself acts as a hapten. When you come to immunology, you know that to elicit an immune response, an antigenic substance must be immunogenic. Hapten is a small molecule that by itself cannot elicit an immune response. It has to be coupled to another bigger molecule, for example, a protein, to induce a certain immunologic response. A third mechanism for cytotoxicity that results from xenobiotics is through chemical carcinogenesis. And this is a figure that illustrates that a xenobiotic through metabolism, for example, through cytochrome P450, will generate reactive metabolites that will generally be hydrolyzed or detoxified to form non-toxic metabolites. If this does not happen, the reactive metabolites can bind covalently to macromolecules such as nucleic acids and proteins, as we have already mentioned, leading to cell injury. The xenobiotic itself will act as a hapten, leading to antibody production and cell injury, here mediated by the immune system itself. The binding can lead to mutation, for example, nucleic acids, leading to cancer. Metabolizing enzymes of xenobiotics are affected by many factors, including genetic factors that are linked to the age and the sex, concomitant intake of inhibitors or inducers, and effect of certain disease conditions such as liver cirrhosis. The fate of xenobiotics in the body is that most of them are subject to metabolism, and the liver is the main organ that is involved in xenobiotic metabolism. The liver possesses more than 30 enzymes dedicated to metabolism of xenobiotics. However, sometimes a xenobiotic may be excreted and changed. There are two main classes of reactions called phase 1 and phase 2 reactions. Both of them aim at increasing water solubility, that is to increase the polarity of xenobiotic substances, and this will facilitate their excretion. If phase 1 met generally, a xenobiotic will undergo a phase 1 reaction which will make it more polar. If this polarity is not enough, then it will undergo a phase 2 reaction to make it more polar. Sometimes phase 1 reaction by itself is enough. It will create enough polarity in the xenobiotic substance. It will be excreted directly without needing to go through a second phase of reaction. Phase 1 reaction occurs first at always. Reactions of phase 1 xenobiotic metabolism are either catalyzed by P450, where hydroxylation is a chief reaction catalyzed by these cytochromes. The reactions of phase 1 can be non-P450 catalyzed, for example, by hydrolysis by esterases. Hydroxylation as a phase 1 reaction is catalyzed by cytochrome P450. Effects of hydroxylation will lead to termination of action in the majority. So an active xenobiotic will be less active. In the minority, hydroxylation will make a substance or xenobiotic more active. For example, ingesting a fruit drug going through phase 1 hydroxylation reaction, it will be an active form of drug. This is a useful part of hydroxylation. However, procarcinogen and sometimes subtypes of procarcinogens, when they undergo phase 1 reaction, instead of being usefully inactivated, for example, they will be more active. They will be converted to carcinogens, which have a harmful effect. So hydroxylation reaction by activation can have both useful and harmful effects. We are going to discuss cytochrome P450. We are sort of going through the CV of this cytochrome. We will talk about its chemical nature, its isoforms, nomenclature, location, function, drug-drug interactions, as well as a couple of examples. The chemical nature of cytochrome P450 is mainly a hemoprotein. So it is a protein that contains heme as a phosphatic group, similar to hemoglobin and myoglobin. Membrane-bound cytochrome P450 enzymes interact with the phospholipid bilayer, particularly with phosphatidylcholine, so it will be linked to lipids. Those lipids are important for the stability of the function of P450, but chemically it is mainly a protein, and in particular a hemoprotein. And there are so many isoforms of cytochrome P450. There is a total of 14 families. 11 of them are detected in humans, and there is at least 150 isoforms. 30 to 60% of them can be present in humans. So there is polymorphism regarding cytochrome P450 presentation within a certain organism or species. Certain polymorphic forms or genetic forms exhibit low catalytic activity, and this will explain variation in drug responses among a certain population, because individuals within a certain population will have different isoforms of P450, so they will catabolize, for example, drugs at different rates, and they will respond differently to different drugs. Regarding the nomenclature of cytochrome P450, it has systemic nomenclature. It exhibits a distinct peak at 450 nanometer, and that is why the name 450. And based on structural homology, CYP stands for cytochrome B450. So it will be followed by an Arabic number, which represents a family, and we say a group of cytochrome P450 isoforms are family when they exhibit more than 50% homology in their amino acid sequence. And it is followed again by a capital letter which represents a subfamily. We say a certain number of cytochrome P450 are a group of the same subfamily when they share more than 55% homology in their amino acid sequence. And then it is followed by an Arabic number which represents the individual number. When it is written in italic, sliding like this, it will be a gene. When it is not italic, it will represent the protein itself. And this is to show you that CYP stands for cytochrome P450. The first number here represents a family. The capital letter represents a subfamily. And the number here represents the number of the member within that family subfamily. Where the cytochrome P450, where is it located? It is present in all tissues, particularly liver and intestine. Within the cell, it is present in the smooth endoplasmic reticulum. In the adrenals, it is present in both endoplasmic reticulum and mitochondria. Regarding the function of cytochrome P450, almost half of the drugs in the body are metabolized by different isoforms of cytochrome P450. So its major function is metabolism of drugs. And cytochrome P450 is considered as the most versatile biocatalyst because it acts on so many substrates. P450 isoforms have overlapping substrate and specificity. The substrates are generally lipophilic, we said. The body needs to resolve the issue of lipophilic being a substance being lipophilic by making it more hydrophilic. Substrates for cytochrome P450 include exogenous substances, including xenobiotics, as well as endogenous substances within the body, including steroids, eicosanoids, fatty acids, and retinoids generated within the body. The exogenous will be taken from outside the body, such as ingestion of drugs. Cytochrome P450 catalyzes a wide range of reactions, including hydroxylation, which is the main type of reaction, deamination, dehalogenation, desulfation, epoxidation, and peroxidation. And the reaction itself is quite complex. It is called hydroxylase cycle. It acts as a mixed function oxygenase or a monooxygenase that incorporates a single molecule of oxygen into the substrate. So R here represents our substrate, and addition of molecular oxygen will lead to formation of hydroxyl group. So this is a hydroxylation reaction. NADPH is used. The other molecule of molecular oxygen will be reduced to water. Drug-drug interaction means the effects of one drug are altered by prior, concurrent, or later administration of another drug. So we say that there is drug interaction when drug A is affected by drug B or vice versa. Whether drug A, for example, was taken prior or before drug B, or the two drugs are taken at the same time, or another drug is later on or administered following another drug. So they don't have to be administered at the same time. It may be preceded or follows it. Still, they can interact because they are still remnants within the body and they are not fully excreted. Some drug substrates have the ability upon repeated administration to induce P450, and vice versa. Other drugs can inhibit cytochrome P450. And these are examples of inducers, and the list here shows the inhibitors of cytochrome P450. This required long list. Inducers of cytochrome P450 include, for example, phenylpropitol, which will induce cytochrome P450, and this will increase metabolism, for example, of chloramphenicol, antibiotic, cortisol, and dioxin are drug used in heart failure. When the metabolism of these drugs will increase, they will be um, they will be needed in more dose, in more larger doses to replace or to have the same pharmacological effect. Rifampicin induces cytochrome P450, which will lead to increased metabolism of covalent anticoagulants such as warfarin, digoxin, detoxin, and the oral contraceptives. So, for example, if a lady is on oral contraceptive and rifampicin is prescribed at the same time concurrently, we need to warn the ladies that rifampicin will lead to increased metabolism of oral contraceptives, which will lead to increased risk or possibility of conception or of pregnancy. Ethanol also increases or induces cytochrome P450, leads to carcinoneutron and nitrosamines activation. So that explains some of the harmful effect of alcohol. Inhibitors of cytochrome P450 include the antifungal ketoconazole, which when administered, it can inhibit cytochrome P450, leading to inhibition of metabolism of terfenadine. And terfenadine is an anti-allergic drug. It will accumulate. It will lead to cardiac problems. Macrolides, antibiotics such as erythromycin and azithromycin, when administered, can inhibit cytochrome P450, leading to accumulation of terfenadine, terfenadine, and methylprednisolone, leading to different pharmacological effects. Grapefruit juice, for example, here is an example of a natural substance, but it can also affect cytochrome P450. It will inhibit it, leading to an accumulation of atorvastatin, cyclosporine, and cisabride. Grapefruit, you need to also warn your patients who are on coumarin anticoagulants such as warfarin to be careful and to avoid grapefruit juice, because grapefruit juice has an inhibitory effect of cytochrome, and it will lead to accumulation of or the same dose of warfarin will be more than the usual, and it can lead to bleeding tendency. An example of cytochrome P450 here is cytochrome 1A1. It is involved in the metabolism of polycyclic aromatic hydrocarbons and related proteins. It is elevated in smokers. Polycyclic aromatic hydrocarbons are inhaled by smoking, and they will be converted to active carcinogens upon the action of cytochrome P450, particularly through hydroxylation. And this explains part of also the harmful effect of smoking. Another example of cytochrome P450 is cytochrome 2C9, which metabolizes the anticoagulant warfarin from the coumarin group. Concurrent use of phenobarbital, an antiepileptic, after 5 days, the PBB hypertrophy, increasing the size of endoplasmic reticulum with elevation of the level of cytochrome P2C9. This will lead to increased metabolism of warfarin, and thus the dose must be adjusted. So you don't have to stop if the drug is needed. For example, if somebody is has epilepsy and he needs to use phenobarbital, and at the same time warfarin will need to be administered. We just need to be more careful with warfarin. Warfarin dose will need to be increased. Another example of cytochrome P450 is cytochrome 2D6, which exhibits polymorphism. It is involved in the metabolism of antihypertensive drug digoxin and spartaline, which is antiarrhythmic. Certain polymorphism in this cytochrome will lead to poor metabolism of both drugs, leading to accumulation and unwanted pharmacologic effects. So the side effects, for example, or the intended pharmacologic effects themselves will be intensified or increased. Phase 2 reaction of xenobiotic metabolism are generally involved in converting a xenobiotic into a more polar form. Phase 2 renders them even more polar than the hydroxylation or phase 1 reaction has made them, and this will facilitate excretion in urine or bile. Phase 2 reaction include conjugation, for example, glucuronic acid with sulfate, with acetate, with glutathione, amino acids, etc. Rarely, conjugation may increase the biological activity of xenobiotics, so it will be more active. Another form of phase or another example of phase 2 reaction is methylation reaction. Phase 2 reaction, glucuronidation, as an example of conjugation, is the most frequent conjugation reaction, where UDP glucuronic acid acts as a glucuronyl donor. So the glucuronyl group will be added to the xenobiotic substance. This will be catalyzed by glucurosynyl transferase. And the substrates can undergo glucuronidation, include benzoic acid, phenols, and many steroids. Glutathione also can be part of phase 2 reaction, particularly through conjugation. And this many potentially toxic electrophilic xenobiotics undergo this type of conjugation phase 2 reaction. The enzyme involved here is glutathione transferase, which is present abundantly in the liver cytosol, but in lower concentrations in other tissues. A third type of conjugation is sulfation, which is a way to detoxify or to deal with alcohols, arylamines, phenols, and steroidal hormones. The enzyme is cytosolic sulfur transferase. The sulfate donor is PAPS, adenosine triphosphate 5-phosphorosulfate, or active sulfate. Acetylation reaction as a form of conjugation where acetyl moiety will be added to the xenobiotic. It is catalyzed by acetyl transferases, which are present in cytosol of liver and other polymorphism in acetyl transferases will categorize individuals as either slow acetylators or fast acetylators. So if you have a population, you can test them for the activity of the acetyl transferase for a certain substance. Some of the people will be slow acetylators, so the process of the reaction of acetylation will be going at a slow rate compared to fast acetylators. The difference is that in slow acetylators, the substance or the substrate will tend to accumulate, while fast acetylators will consume the substance more fast or acetylate the substance more fast, making it more polar, and it will be excreted easily. So there is no accumulation. If a drug is the same drug is administered to a person who is a slow acetylator and to another person who is a fast acetylator, the slow acetylator is likely to give the toxic effects of a certain substance due to accumulation of substance. And an example of this substance is isoniazide, which will have toxic effects in slow acetylators who fail to acetylate isoniazide at a normal rate. Methylation reaction. Few xenobiotics are subject to this type of phase 2 reaction. It is catalyzed by methyl transferases, where S-adenosine methionine acts as a donor of the methyl group. And this concludes our report. Thank you very much.